Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in this video I am going to show you the solutions to paper 4 May-June 2019. So for those of you who may just not be aware, or if you've just found this channel has your biology syllabus for the Cambridge AS and A-Level Biology in chronological order. So if you go to the playlist function, you will see that everything has been arranged by chapters. And what I'm going to be doing once all the chapters have been completed would be to post more of these types of videos where I am solving past papers to sort of help you see how to just get the solutions to past questions. So I hope that you find these videos helpful, and I hope that you're encouraging your colleagues to use them as well. For those of you who are going to be writing your CAIE exams this year in May, June, so basically by next week, I just want to say that I wish you good luck, and I'm going to give you some information about paper 4. So paper 4 is always for 100 marks when you take the CAIE exams. So it's for 100 marks and you have 120 minutes to do it. So that's 2 hours. And so what that tells you is that per mark, you have 1.2 minutes per mark. So if you have a 6-mark question, you just need to multiply 6 by 1.2 to get the amount of time you expected to sort of spend on it. But obviously, I know the students don't do that. You typically just answer the questions that you can. So, hey, just go for it. But I just want you to know that with this particular paper, you should be very alert with time and try as much as possible to answer the questions that you know as soon as you see them, rather than sort of procrastinating. So usually I tell students to just start with what they know, and then they can dwell on what they're not really sure of. Something else to tell you about paper 4 is that it counts 38.5% towards your final grade in CIE. 38.5, it's the highest aggregate for all of the papers that you would write, or rather the highest contributor to your aggregate score at the end of the exam. But usually if you're able to get about 75, 78% or 80% in this paper, you should be fine. You should be able to get an A actually. So by all means, just work hard. Paper 4 covers the chapters from chapter 12 to chapter 19 of the biology syllabus. So you're going to be preparing for that if you're writing next week. You're writing on the 5th of May. So just focus your energy on solving papers and questions that are all basically paper 4, so that you're able to see the style of how questions are asked and you sort of know how to answer. In this video, I expect it to be a bit long because I'm sort of solving most of the paper from 2019. So please, if you feel like you need to pause it at some point, get some water, get something to eat, feel free to do so, but don't disregard any of the answers or the questions or the parts, because I will be explaining concepts as I go along as well. I haven't seen the questions before, so I expect that as I just go on, we will solve them together, and hopefully I don't make any mistakes, but well, we'll see what happens. Okay, so this is the first question here. It says figure 1.1 shows part of a cell in the growing region of a plant. So you know you're dealing with a plant cell, and it shows you there the cell wall. So I'm going to be annotating using my red pen as usual. So there's the cell wall, and then this is the cell membrane over here, which already again tells you this is a plant cell. And it says here, state the type of protein represented by A. So here you just need to come in and you look at A. So what is A doing? A is accepting an auxin. This molecule is an auxin. And so in this case, A would be an auxin receptor over there. And then proteins B, C, and D are transport proteins. Identify proteins B, C, and D. So let's have a look at what B is doing. What is going through B? This is a proton, right? And so that tells us that B is a proton pump. And over here we have potassium coming in, and also we can see that this is split in the middle, so it sort of has two parts, so it's like a gate, and this then can tell us that this is a potassium channel protein, or potassium ion channel basically. And then we have D as well, and D is very similar to C, but what is passing through D is water. And so in this case, because we know that we're dealing with a plant cell, and the only things that tend to pass, like the only time water would pass through a specific channel is if that channel is an aquaporin. So this over here, auxin receptor, I'm just going to write receptor. Okay, and over here proteins B, that's a proton pump, so a pro pump. I'm just going to put that there. This mouse I use these days is not the best. And we have a potassium ion channel pore, or a potassium channel would also be fine, by the way. And this here would be an aquaporin. All right. And you have to get that right, by the way. You need to say it's an aquaporin because it has water specifically traveling through it. Okay, and then B says, describe the effects on the cell wall of many hydrogen ions moving into the cell wall. Now, this is asking a question about how plants grow, and I think this is something to do with coordination, if I remember correctly, where we discussed how the cell wall expands in response to the pump, in response to more hydrogen ions being pumped into it. So what you need to then say here is that when hydrogen ions, you can read this just to see how auxin stimulates stem elongation. I think that was the question. It's either coordination in plants. It has to be coordination in plants. That's chapter 15. So if you haven't seen the video, make sure you just go and check it out because that would be very helpful for you. But when protons are pumped into the cell wall, what happens that you might be able to guess is that because of the way they are, they make the cell wall more acidic. And when they make the cell wall more acidic, they loosen the cellulose linkages that are within the cell wall. And as a result of that, you would find that expansines, there are these proteins called expansines that are secreted in response to that. So that then causes the cell wall to expand and sort of leads to elongation of the plant. So that's just one way of putting it, but this is for 3 marks. And I just want to say that whenever you have the number of marks written out, try to write, especially if it's a question like this, just try to write 3 key points because those are the 3 you would get marks for anyway. Obviously, you can go on and write up to 5 as long as you don't exceed the space that has been provided, but you would only get 3 marks. So sort of be clear on your answers. And when it says describe means that you need to sort of tell it like a story, like you're telling what would happen. So again, if you pump protons rather, into the cell wall will become acidic. And when the cell wall becomes acidic, that secretes the production of, that activates rather the expansines in the cell wall. The expansines will cause the loosening of the linkages between the cellulose fibers and the cell wall. As a result of that, water will move into the cellulose fibers and that will cause an elongation of the cell wall of the plant and result in elongation of the plant itself. Okay, let's look at the next question. So over here, I didn't put, that's not the whole question for question 1, by the way, but like I said, I'm only solving most of the paper. So if there's any particular question in the paper that you're just like, oh, she didn't solve this and I would like some clarification, you can sort of type it in the chat and in the comments and see if someone from the learning community can get back to you, or if I can, then I would. Okay, so over here it says the interpupillary distance is the distance in millimeters between the centers of the pupils of the eyes. And then figure 2.1 shows us how the IPD is measured. So you can see over there. And IPD is one example of a characteristic of human facial structure that shows variation. So obviously when we're dealing with variation, we know that we are now in the range of chapter 17. But something I have to tell you, and I tell my students this, is that when you go into CAIE and you see a question, you can associate it with the chapter, but make sure that association is really loose because you might find that chapters overlap in certain questions so that you don't end up giving the wrong answers because you really believe that it has to come from that chapter only. All right, so figure 2.2 shows the pattern of variation in IPD in a sample of adults. So let's see what the question is. Name the type of variation shown in figure 2.2. So always remember there are 2 types of variation. You can have continuous variation. Continuous variation is usually non-distinct and it's normally distributed for the most part. And you would also find that it's something that sort of varies over a range. Whereas discontinuous variation is variation that puts things in categories, so very distinct categories. So an example of discontinuous variation rather would be something like blood groups. So there's no range between A and B in blood groups, for example. So people are not like A1, A2, 8.5. That's not how it works. So you're either A or you're B, right? But with things like this, where you're measuring distance, and just you can see here, there's a range, because people sort of starting from maybe like 51, 52, 53, 54, 55, and then they go on. That tells you that there is a range. There are no distinct categories, and as a result of that, this is continuous variation. Suggest and explain how genes and the environment contribute to variation in IPD in humans. So when it comes to continuous variation, you always know that there are many genes that affect that characteristic or that trait, and all of these genes have an additive effect and they interact with each other. Also, you can say things like the environment can affect gene expression, for example, diet, and we covered this on the slides, by the way. So if you haven't watched the videos on chapter 17, this is a good time to go and do so. But things like diet, access to nutrition, and all of those things can affect can affect the variation or can contribute to the expression of certain traits in organisms. So let's look at B part of that question. Individuals with an IPD of 70 millimeters or more have a mutation in the PAX3 gene that results in less PAX3 protein being made. The normal role of the PAX3 protein is to increase the expression of many other genes involved in embryonic development. These genes affect a range of phenotypic features such as facial structure, hearing, and eye color. State the term that is used to describe a gene such as PAX3 that controls the expression of other genes, and suggest how the PAX3 protein controls the expression of other genes. So this is a chapter 16 question. So if you haven't watched the last video in chapter 16 where I discussed gene control, lac operon, and so on, then this is a good time to do so. Again, I hope these questions are sort of bringing up to you the areas where you might not really be clear, and you would go and do like some really hectic revision after watching this video. But the PAX3 protein here is being said to be the protein that controls the expression of other genes that affect embryonic development. What this means is that the PAX3 gene is a regulatory gene. So regulatory genes are genes that sort of control the production of transcription factors for other genes. And when a regulatory gene sort of switches off a set of genes, those genes will not be expressed. So a regulatory gene is certainly what you'd call the PAX3 protein, because that's what it does. It controls the expression of other genes. And so over here, the way it would do that is that it would sort of control the transcription factors. So it would control like the activators or the inducers and all of those things. And what it would then do is that when it produces an inducer or an activator, that activator is able to bind to the DNA of the other genes and transcribe them into RNA. And as a result of that, cause the expression of the genes. A regulatory gene also stimulates the activity of RNA polymerase, so that is another way to look at it. Let's look at the B2 part. It says, describe how microarray analysis could be used to identify the genes switched on by PAX3 in embryonic cells. So I just want to say that with microarrays, you are usually using them to check for the expression of genes, for genes that are being expressed. And the way you can do that is you can get the mRNA from the cells that are in in the organism. So you can get the mRNA, and when you get the mRNA, you make cDNA from the mRNA. So basically you reverse transcribe it from RNA to DNA. You add a fluorescent tag to it, and then you add these to the microarray tray. And when you add this to the microarray tray, the cDNA would be able to bind with complementary bases in the microarray tray. And wherever you have your fluorescent indications, you can then tell. So say, for example, you label the genes from PAX3 as maybe yellow, and the complementary base pairs in the tray are perhaps maybe red, for example. And so wherever the PAX3 is able to bind with a complementary base pair, it might give a different tag, maybe green, because of the combination of red and yellow. Obviously, I'm not saying that that's the way red and yellow combine. They combine to form orange, but I'm just using it as an example. But wherever there's no binding, you would find that the microarray would just be yellow, because that's what you've tagged your PAX3 mRNA as, or your PAX3 cDNA as. And in places where the Microarrays just on its own, you would just have red. So if you haven't, again, this microarrays come from chapter 18, which I know I haven't done videos on chapter 19, sorry, which I haven't done any videos on, but I really, really hope that you're able to at least get to it before you have to write your exams on the 5th of May. Okay, let's look at B3. Then it says here the chimpanzee Pantroglodytes has DNA that is 98.5% similar to humans, including possession of the PAX3 gene. Investigations show that chimpanzees express higher levels of the PAX3 protein during embryonic development than humans. And so suggest how knowledge of the PAX3 gene helps scientists explain how humans and chimpanzees are very different in facial structure, even though they have similar DNA. So in this case, you can talk about how, because again, it says suggest, right? So you're not thinking about facts from your textbook, you're thinking of the application of the information you've gained in biology. So an example here would then be to say that there might be a small genetic difference in the PAX3 sequence in humans and chimpanzees that results in them having, these differences. So just one small change in alleles can result in big phenotypic differences. Because the PAX3 controls so many other genes, it might then have a different effect in chimpanzees than it would have in humans. And also the fact that the PAX3 gene is higher in concentration in chimpanzees might then be the result for their eyes being close together, because remember it said when we were reading the question earlier that the more PAX3, like the PAX3 gene controls distance between the eyes. So you can look at this chimpanzee. The image is also there for you to observe and sort of make an inference on what you might put as your answer. But sometimes suggest questions can be quite difficult for students, but by all means think about it and just think of how you would apply what you have learned and what you've learned also from the question, because most of the time the answers are hidden in the question. Okay, this one looks a little bit small, but I hope that we can see it. But I'm just going to read it out anyway. So it says here, some neurons in the brain produce a neurotransmitter known as dopamine. I'm sure you've heard of dopamine before. And it says Parkinson's disease occurs when the neurons that produce dopamine die. A person with the disease may experience difficulty in coordinating movement, often seen as tremors in different parts of the body. Parkinson's disease typically occurs in people older than 55 years. Younger people with these symptoms are said to have early onset Parkinson's disease. Recessive mutations in a gene known as PINK1, located on chromosome 1 and autosome, are believed to be one cause of EOPD, so that's early onset Parkinson's disease. A person with this form of EOPD has a homozygous recessive genotype. Draw a genetic diagram of a cross between two individuals who are heterozygous at the pink locus. So in this case, there are lots of buzzwords that you just need to take note of. Over here, I tell students because I've noticed that with many biology students, whenever they come across a question that looks difficult or looks detailed, they become really scared and they just sort of mess things up. So I'm going to split this question into parts for you just so that you can see that there's absolutely nothing scary about it. Over here, from here to here, it's just information, okay? This is information that's being given to you about how Parkinson's would affect people typically, but it doesn't have anything to do with the biology that you are about to solve. It might become applicable later, but for this question A I'm looking at, it doesn't. Question A requires you to focus over here, where it says recessive mutations in a gene. So you know that Parkinson's is the result of a recessive mutation, is believed to be one of the causes of EOPD. And a person with this form of EOPD has a homozygous recessive. Now remember what we said that when you write a dominant allele, you write it in capital letters, and when you write a recessive allele, you write it in small letters. If a person is homozygous recessive, it means that they have two recessive alleles, okay? And if a person is homozygous dominant, then they have two dominant alleles, like that. So I hope that makes sense. If we say that a person is heterozygous over here, then it means that that person has one dominant and one recessive. That is what it means. So with that in mind, let's then look at what we are going to do here. So we're supposed to draw a genetic diagram of a cross between two individuals who are heterozygous. And so what we can do here in this case is that we can choose A as our dominant and small a, big A as our dominant and small a as our recessive. And so in this case, you just need to say that the normal allele, because the normal one is the dominant one, so you can say A is equal to normal. And make sure you write it out, because another thing students do that I find to be a very silly oversight on their part is that they would write the symbols but not tell the examiner what it is. So you might say, okay, well, it's general knowledge that the dominant allele is always written as a capital letter, but what if you have chosen differently in this case? You need to make sure that you let your examiner know exactly what you are doing. And so in this case, small a would be equal to recessive or mutation, all right, mutated allele. So I'm just going to write it that way and leave it at that. Now for the parental genotypes, because we've been told that these two individuals who are being crossed are heterozygous, so that means they are A, all right, times big A, small a as well. Now, something I need to say to you is that in this case, you should always know that when it comes to doing these crosses, because over here you can see it just looks like you don't have any room, but you can always sort of scribble somewhere on the corner of your paper. You'll notice in your CIE question paper, when you're going through it, that you have blank pages. So you can use those blank pages and a pencil to sort of work out things that might not be clear to you. But these are the parental genotypes, and in this case, the gametes would be simply a, big A, all right, and small a. This mouse I use is not the most user-friendly mouse, and then big A and small a. So basically these are the gametes, because those are the only things that you can get when you cross these two together. If, for example, we were crossing big A times big A, small a, then our gametes would be very different, because we'll then have 3 big A's, and we would have 1 big A, 1 small a, 2 big A's, and so on. So basically, I hope you get that. All right, let's go to the next question. It says, what are the offspring genotypes? So over here, you might need to do, just to show you how this would look so that you don't make any errors. Oof, that wasn't a good one. It's just so that you don't make any errors. I'm just going to show you what it should be. So over here we would have our big A, and we would have our small a, and then we have big A here and small a there. And please know that in the CIE, even when you're answering the questions, it's okay for you to write it like this. You don't, if you know you're going to get confused or you can't really predict, you're not sure, by all means do this so that you are very clear on what it is exactly that you're doing. So this is big A, because we're crossing that with that. And then when we cross these two, we're going to have big A, small a. And then for these two, we're going to have big A, small a again. And over here, we're going to have small a, small a. All right. So we have 1 homozygous dominant, we have 2 heterozygous, and we have 1 homozygous recessive. Those are the offspring genotypes. So once you've done the cross like this, make sure you come and you write it out here. So you write AA like that, and then you write AA, okay? And then you also write AA again. My mouse is giving me problems here, but I think I need to change the mouse pad I'm using. And then we have small a, small a. So once you have those, you need to make sure you write them out like this. And then what now is the ratio of offspring phenotypes? Now this is something where students also tend to make mistakes, because they tend to assume, for some reason, that the question is asking them for genotypes. If the question is asking you for genotypes, in this case, the ratio is 1, which is homozygous dominant here, 2, heterozygous would be 2, and homozygous recessive would be 1. That's the genotype. The phenotype, on the other hand, is asking you about the expression of the gene. Like, what would this person look like, or what traits would they express based on their genotype? So if we look at the offspring genotypes that we've got over here, this would be a normal offspring, because this offspring doesn't have any recessive traits which would cause early onset Parkinson's disease. This here that are heterozygous, because they have the dominant normal allele, they would also be normal. All right, so they would also come out as normal. And over here, this one would have early onset Parkinson's disease simply because it has two recessive alleles, and those recessive alleles are responsible. So the ratio of offspring phenotypes, the phenotypes would then be 3:1. And that is how you solve that question. Okay, let's look at the next question, and this is question 4 in the question paper. So again, I haven't done every single question because I believe that some of them you might be able to just get through, but I've chosen most of the questions from the paper. All right, let's look at this one. It says the link reaction and Krebs cycle take place in the mitochondrion. The main stages of the link reaction and Krebs cycle are listed in table 4.1. They are not listed in the correct order. So already, before even looking at what the question says, you can already tell that there's a high possibility that you will be asked to recall the steps in respiration. And not just all the steps of respiration, but specifically what happens within the link reaction and the Krebs cycle. I love questions like this because they sort of test your ability to recall or synthesize information in a chronological order. And you might already tell from this channel that I love chronological order. So let's look at it. So let's see what the question says actually. It says complete table 4.2 to show the correct order of the stages. 3 of the stages have been done for you. So over here, they've already told us that E is number 1. So what I would advise students to do here is to just come in and put the numbers, all right? E is number 1, J is number 6. Okay, and D is number 11. Okay. All right, so we know that, and now we are going to put in our own numbers, just to see what it should be like. So over here, let's see. It says E is number 1. Pyruvate enters the mitochondrial matrix. So that sounds like the beginning of the link reaction. When the link reaction happens, this is a chapter 12 question, I believe. When the link reaction happens, when pyruvate gets into the matrix of the mitochondria, what happens there is it is decarboxylated, which means it loses a carbon in the form of CO2, and it loses a hydrogen as well. And that is why during the link reaction you form NADH. It loses a hydrogen as well, and it becomes acetal. Acetal then combines with acetyl coenzyme A, and that is what the coenzyme A helps acetal get into the Krebs cycle. So that is just the link reaction in detail. So let's see which of these tell us about that. So let's see, pyruvate enters the mitochondrial matrix. Acetal group combines with, so now I'm reading from the top. Acetal group combines with coenzyme A to form acetyl-CoA. Can't be too sure. Citrate is formed. This is from the Krebs cycle. Hydrogen atoms are accepted by NAD and FAD. That is from the Krebs cycle. Oxaloacetate is regenerated. We've been told that's even the last step. Acetyl group is formed. Acetyl-CoA enters the Krebs cycle. So we know that G is definitely going to come after A, right? We know that G comes after A. And then over here, let's see, ATP is made by substrate-linked phosphorylation. There is no ATP made during the link reaction, so we know that that's not possible. Pyruvate is decarboxylated and dehydrogenated. This certainly has to be 2. Based on what we can see, citrate doesn't play any role in the link reaction. So pyruvate is decarboxylated and dehydrogenated, and once that happens, we're going to form an acetyl group. So this then becomes 3. Okay, and when that acetyl group is formed, the acetyl group will combine with coenzyme A to form acetyl-CoA. So this is 4. Acetyl-CoA will enter the Krebs cycle. This is 5. Okay, so we've just done the link reaction, all right? Acetyl-CoA enters the Krebs cycle. When acetyl-CoA enters the Krebs cycle, we've already been told that this is 6 over here. So 6 is acetyl-CoA will combine with oxaloacetate, and it would form citrate. So this has to be 7 because it says here citrate is formed. So this has to be 7. And let's see what happens next. Citrate, what do we have left? We have hydrogen ions accepted, oxaloacetate, ATP is made. So over here, citrate is decarboxylated and dehydrogenated. For me, that would be 8. Okay, and then it says, so if we're removing the hydrogen from citrate, those hydrogen ions that we remove end up going to NAD and FAD in the Krebs cycle. So this has to be 9. And because we have only one thing left, then obviously this will be 10. And so in this case, once we have done this, and the usefulness of doing this first before sort of stressing about what to put in the box is that now you can see clearly what your answers should be. So number 2 is I, and so we can just put I there. And number 3 is F. And of course, you can check if this is correct with the mark scheme. So if I'm wrong, by all means, please post it in the comments. I do not mind being corrected at all. And then A would be number 4 over there. And 5, I believe we said G comes after A, so that would be G. And we know that J is our number 6 over there. Where is number 7? 7 is B. So this would be JB. And then 8, where did we say 8 is? K. So that would be K. And then 9, we said the hydrogen atoms would be accepted, so that would be C. And so 10 would be H. And this is how you answer these kinds of questions. Very easy and easier than you trying to like go from there back and forth. Just look at the sequence of events and solve it on the one side before you move to the next. All right. Here then it asks us what the role of NAD is in aerobic conditions. I deliberately chose this question because I realized that many students don't understand what NAD does. Like they know that it is a hydrogen carrier, but they're not really sure of what it is. And also sometimes when students are asked in CAIE questions to identify what a coenzyme is, because they don't see coenzyme A, they see NAD, they tend to not pick NAD. NAD is actually a coenzyme, and what it does is that it aids, it accepts hydrogen from dehydrogenation, and it carries that hydrogen to the electron transport chain where ATP is produced. And so if you're able to sort of list those things, you would be able to get full marks here. So I just thought to highlight that to you. Then over here we have a question that speaks about carbon dioxide is removed from compounds in the link reaction and Krebs cycle by decarboxylation. So let's look at this. Something to bear in mind when you are looking at the link reaction and the Krebs cycle is that the link reaction has a decarboxylation step. So I'm just going to try to write it out to explain what the answer to this question here should be. So you have pyruvates coming in. I'm just going to write it as PYR. It's coming in, and over here one CO2 is removed. Okay, and that pyruvate becomes acetal, and acetal combines with CoA and goes into the Krebs cycle. And when it gets into the Krebs cycle, it is then it loses 2 more carbons actually, or rather the whole process. There are 2 more carbon dioxide molecules that are lost. So that means we have CO2 times 3 that's lost in the link reaction and the Krebs cycle. But that is not where your answer stops. So the answer here is not 3. Remember that for you to get pyruvate, you have 1 glucose molecule from glycolysis. And that glucose molecule is split into 2 molecules of pyruvate. Okay, so that means that each pyruvate molecule that goes through the link reaction will yield 1 CO2, right? That's for sure. But as it goes on through the Krebs cycle, it will yield 2 extra CO2, so that's 3. And because you have 2 pyruvate molecules from 1 glucose, then the answer here should be 6. Then it goes on, so that's just the answer to question 1. Then in question 2, it says, in the mammal, carbon dioxide diffuses from cells in the blood to be transported to the lungs. Suggests while CO2 is transported in the blood, mainly as hydrogen carbonate and not as carbonic acid. Now, this is something that typically you would find in your chapter 8 from your AS biology syllabus. And I know that for so many students, they always like it's so unfair that sometimes you'd be doing paper 4 and you've solely focused on studying the last 7 chapters, but now here comes a chapter from the AS content. So again, just try as much as possible to glance through your AS, but study your A level in deep detail, and not just study to remember, but sort of study to be able to apply the knowledge as well. So the reason why we transport CO2 in the blood mainly as hydrogen carbonate and not as carbonic acid is that carbonic acid would dissociate when it's in the blood. So it dissociates because it is catalyzed by, the breakdown of it is catalyzed by an enzyme called carbonic anhydrase. You can also say that the hydrogen carbonate ions prevent the decrease in blood pH, so they act as a buffer. So if you haven't looked at any of the chapter 8 videos, please by all means do so. Okay, so I hope this is clear because I can see that the letters are a little bit small, but let's just try to get through it. I was trying as much as possible to fit the information as clearly as I could, or most of it onto the slide. So researchers investigated the extent to which the founder effect and natural selection affected evolutionary change. So here, these are your buzzwords: founder effect, natural selection. Figure 5.1 shows the brown Anole lizard and Anole Asagre. These lizards live on a number of Caribbean islands and feed on a variety of invertebrates and other small animals. And so we can see there's a lizard over there. Asagre rather spends lots of time perching, so resting on, moving along branches of shrubs and trees. The width of the branch that it perches on is known as the perch diameter and is labeled in figure 5.1. There is a positive correlation between perch diameter and hind limb length of the lizard. Longer hind limbs show the lizard runs faster in vegetation with a larger diameter, and shorter hind limbs are needed to provide stability on vegetation of a smaller diameter. I hope we'll be able to remember all these details. If you can't, and we feel like this is just too much information, a suggestion would be at this point to maybe find this question paper, download it so that you can sort of look back when we're answering the questions. In 2004, I'm also taking notes here just so that you know. In 2004, a hurricane caused the death of all the lizards on 7 islands. In 2005, the researchers randomly selected 7 male and 7 female lizards from a source population on a nearby island. For each of the 7 islands affected by the hurricane, a male and female lizard were mated and placed on each island. These islands formed the experimental founder islands, where new populations of the lizard were successfully established from each founding pair. And then figure 5.2 shows the difference in vegetation between the source island and the 7 experimental founder islands. So this is the source population, and this is the experimental founder island. So I'm just going to summarize what this story has been so far. So we have this lizard here, and they found on this species of lizard is usually found on a number of Caribbean islands, and it perches on certain kinds of vegetation, and that perch, the kind of vegetation it perches on, has a certain width. So you can tell by its perch diameter, so by the diameter of the tree branch or of the grass or whatever it is that it's perching on, if the lizard has a longer hind limb or if it has a shorter hind limb, because it's sort of affected by that. There is a positive correlation between the perch diameter and the hind limb length. So if it has a longer hind limb, it is able to run faster on vegetation that has a larger diameter, and if it has a shorter hind limb, it is able to be, it's stable on vegetation of a smaller diameter. And so after a while, a hurricane came and destroyed all these lizards on 7 islands. And in order to re-establish the population of lizards, scientists went to a nearby island, and they mated a male and a female from those islands. This male and female are not the same species as the lizards that were destroyed, but they mated them and sort of put them in the 7 islands that were affected to re-establish the population. And what we can see here is that those islands where the male and female were taken from have bigger trees, if you want to think about it that way, whereas with the founder islands that the scientists were trying to populate, the vegetation looked a little bit smaller. So that's just to summarize what this story is about. Okay, so let's look at the first question. It says, predict the effect of natural selection on mean hind limb length. of Asagre on the 7 experimental founder islands. So if you remember from the image we just saw of the experimental founder islands, the vegetation there looks smaller. And we sort of said that, or something that was given to us in the question rather, is that the mean hind limb or the hind limb length is usually shorter in the lizards that need it for stability, and stability would be required on shorter vegetation. And so in this case, we would then be able to say that the mean hind limb length would decrease because the legs of the lizard would get shorter. So that would be a good way to predict what the natural selection effect would be, because again, natural selection is basically selecting for individuals that are best suited to survive in a certain kind of environment. And so if the environment is filled with vegetation whereby the lizards need short hind limbs in order to survive so that they can be stable on vegetation, then those are the lizards that will be selected in favor of, and so they are the ones that are likely to survive while the others are likely to die off. The ones with the longer hind limbs are likely to die off because they would not be able to stay stable. All right, then predict how collecting individuals at random, and always notice, so something else about CAIE that I tell students, notice the letters that are, or the words that are put in bold, because they mean something. They're trying to call your attention to it. Predict how collecting individuals at random for the 7 founding pairs affects the mean hind limb length of the lizards on the different islands. Now, when you collect individuals at random, something to remember is that that increases their allele frequency. And if allele frequency is increased, then that means there'll be a high variation. And so here you can simply say that the mean hind limb length would have a high variation, or it will vary, and that should give you 1 mark there. So what I've done here, I'm going to show you the next question, but I've just sort of put this image here so that it's a bit clearer, because it was looking a little bit tiny when I was reading it earlier, or trying to record the slide earlier. So many generations of the lizard were produced over the 4 years after the introduction of the founding pairs. So the founding pairs were basically lizards that were reintroduced into these islands that had been destroyed, and they then repopulated the island. And it says figure 5.3 shows the mean hind limb length of the lizards shows how the mean hind limb length changed on the 7 experimental islands and on the source island. So first things first, let's look at the source island. That's the circle. So I think that should be the source island over there, right? It has like it's a circle with a cross inside of it. And then island 1, you can see there's a decrease, and basically all of them are decreasing. That's something that you can just sort of glean from looking at it at first glance. So then the question says, with reference to figure 5.2 and 5.3, describe and suggest explanations for the results for the islands. So 5.2 was the earlier image we saw, whereby we had the vegetation from the source island being a big tree, and the vegetation from the founding islands or the experimental islands being quite short shrub-like vegetation. So in this case, what you can do is that you can sort of describe, so it says to describe and to suggest. So always remember, because look at that, it's actually underlined. So it means that 1 point that you give, you have to give 5 different points, but each point you give has to have a description and a suggestion. So I think my first description would be that if you look at the graph, when you're describing, you're looking at the graph, okay? That's how you describe. So describing shows you that on all the experimental islands, there is a decrease in the mean hind limb length. You can see that from year to year. So it starts off a little bit high and then it continues to decrease. And the explanation for this would then be that there is a selection pressure that is causing this decrease in length, because the animals need to sort of have shorter hind limbs in order to survive. That kind of selection that sort of selects towards a specific characteristic is called directional selection. If you also look at the source population, which is the one at the top over here, so I'm just going to circle it. Oops, that's not a great circle, but let's look over here. If you look at this top here, the mean hind limb length stays the same. Nothing changed in that graph. And that is some kind of stabilizing selection. So that's also something else that you would want to just highlight. And then you can sort of talk about the selection pressure. So you can speak about, so again, it says describe and suggest. So if you've described and you've suggested, you've described the overall length. Something else I would say to you here as a student is that if you're not given any values, so over here, there are no values for mean hind limb length. If you look at it, it just tells you that there is a general trend of decrease. Please don't make up values. Don't come here and say, oh, if this is 0, then this must be 4, this must be 8, this must be 10. Don't do that. If you're not giving values, it means that you don't need to make reference to any values. All that you need to do is simply look at the general trend. All right, so don't make up values for values that you haven't been given. So talk about the selection pressure. Talk about the fact that when you have selection pressure on specific alleles, you would find that some alleles would die off. So for example, in this case, the lizards with longer hind limbs would tend to die off, while the ones with shorter ones would survive based on the way the vegetation of the area looks, or of the island looks. And the ones that survive are the ones that are able to pass their alleles to the next generation. And as a result of that, over time, you would find that you will only find lizards with shorter limb lengths on that island. I hope that was clear, but those are sort of some of the ways you can answer this question. And again, whenever it says with reference to figure 5.2 and 5.3, always make sure to stay within the references. Don't go around sussing for information where you're not asked to look for it. Okay, and then on this question, it says, in the investigation, one population was established on each experimental island. Outline how speciation may occur on the 7 experimental founder islands. So again, when you think about what is going on here, we are actually dealing with a situation whereby 7 different pairs of lizards are being put on 7 different islands, and the hope is that they would establish a new population of Asagre. Now, in this case, because they are separated by geography, that would be called allopatric speciation. If you haven't watched the last video of chapter 17, which I uploaded a few days ago, please make sure you do so, because allopatric speciation is explained there. And so because the lizards are on different islands and geographically isolated, they would have different selection pressures, and as a result of that, different mutations will also occur. And the lizards will sort of evolve differently from each other, and they might not even be able to mate with each other again. So that then results in different species of lizards on the different 7 islands. Then again, here it says speciation is one possible outcome for the experimental founder populations, but there is also a higher risk that they may become extinct. Explain why the experimental founder populations are at high risk of extinction. Now, extinction is something that would happen whenever you have small populations. It's easy for you to throw a small population into extinction. Also because you only have 2 founding pairs that were put on the island. So those 2 founding pairs are the ones that carry the alleles, and they're the ones that pass them on. And so because of that, you have a very low gene pool, or a very small gene pool. And that just means that you don't have enough gene flow that would then result in variation in the population. So if there is less variation, then it means that if a selection pressure comes, it's likely to wipe out the entire population of lizards. Also, here, again, just want to highlight that whenever you have like 3 marks, just writing 3 different points is enough. You don't have to write stories. And also remember to write 1 point per line, all right? Or you can write 1 point from here to here, another point from there to there, another point from there to there, just to make your work neat and easy for the examiner to read. Something else might be that if there is a disastrous event, then all the lizards will be wiped out. Okay, let's look at another question. So this question, I believe, is the 6th question in the paper. And it says maize, maize, and that's the scientific name for maize, by the way, is an important food crop for human consumption and for feeding to animals. 2 variations of maize are Mon 810 and Justina. Well, that's a weird name to call a maize, but let's go on. Justina has been developed in the traditional way by selective breeding, which is artificial selection. You can always watch the video on artificial selection if you haven't. And Mon 810 is an example of a genetically modified organism. This is chapter 18. That's where you discuss genetic modifications. Mon 810 produces a chemical that is toxic to insect pests. It is described as insect resistant. Outline how genetic engineering gave MON810 the trait of insect resistance. What this question is simply asking you to do is to sort of explain how you would transform this maize into an insect-resistant maize. And in this case, you just need to outline the genetic engineering process, which means that you can take the gene from species that is resistant to insects. You cut out the gene using a restriction enzyme. You insert the gene using a plasmid into the nucleus of the plant, which is MON810, and as a result of that, you get your new DNA that then expresses this trait. So you also need to make mention, because when you're discussing the genetic engineering process, that you use marker genes to identify if the gene transfer has been successful. In this case, it says outline, so you don't need to write stories, you just give them bullet points and you would get your answer. You only need to write 4 different points in order for you to get full marks, but if you know more than that, by all means do so. But 4 points should be enough if you're confident in your answer. Then let's look at this. It says countries vary in the extent to which they grow GM varieties such as MON810 instead of traditional varieties such as Justina. In the USA, 88% of the total maize that is grown is genetically modified. In most European countries, 0% of the maize that is grown is genetically modified. Scientists use computer models to predict the effect of 2 different changes in agricultural practice on maize crop yields. So one change is a global ban that reduces the cultivation of GM maize to 0% everywhere. And the other change is all countries increasing the cultivation of GM maize to 88% to the 88% level of the USA. Then it goes on to say table 6.1 shows the results of this modeling for 4 countries. And you can see there on table 6.1 what the different values are. But let's see what the question says. Explain why the data in table 6.1 suggests about the social and ethical implications of growing GM maize. Now, this is interesting because usually when questions come about in genetic modification, that's in chapter 19, I hardly see questions on the ethics and social implications, but I get my students to do presentations on these just so that they can sort of wrestle with the ideas of ethics in biology. But in this case over here where you just need to look at the numbers, okay? So it says explain what the data in table 6.1. It doesn't say give your opinions or feelings about genetically modified foods. So don't come in and say, oh, well, I feel like this is going to result in people getting sick or getting or dying. That's not what you need to look at. You need to come and look at the data. So here the data is telling us this is the percentage change in the yield of maize, and then it says when you decrease GM maize to 0%, that's change number 1 over here. There is an overall decrease in the amount of maize that is produced. So there's an overall decrease in yield of maize. If you increase GM maize to 88%, there is an overall increase. For the USA, obviously, the increase would be 0 because the USA is already at 88%, okay? And then you can sort of suggest, based on this data, that growing GM maize would increase food supply because GM maize would basically increase the overall yield, so that increases food supply. Now again, I think this might be a little bit unfair to many students who are not widely read, but if you increase the supply of maize in the countries where you do GM maize, what that would do is that it would decrease the cost of food. So you can say that then decreases the cost of food for people. And if it decreases the cost of food for people, it means that more people are well fed. So that would be one example. So again, over here, you only have 4 marks. So you don't need to write too many stories or don't write an essay. Just write the 4 key points that you need to do. So point number 1 would be that you have a decrease in food for places where GMAs goes down to 0, and you have an increase where there's GMAs increase to 88%. Then you would also say that there's a decrease in the cost of food because of the increase in GMA supply, and also you can also mention that there is an increase in GMA supply. That would then also help you. Then GMA supply would Keep a country would help a country feed its hungry population. That would also be another mark. And you can also zoom in if you want to. Like, for example, if you look here, the Honduras Honduras would be the country that benefits the most from GM maize cultivation. So you can sort of zoom in there, right? That it benefits the most compared to the other countries. And so that might be a good thing for the Honduras country. And that should give you 4 full marks. Now let's look at another question. I believe this is a different question because I see it says A here, so I'm guessing this is question 7. And it says here, some people have a condition called diabetes. In type 1 diabetes, the pancreas does not produce enough insulin. Figure 7.1 shows the blood glucose concentration of a type 1 diabetic person and a non-diabetic person at regular intervals after a glucose drink. So if you look here for a non-diabetic person, they have a glucose drink taken here at 0. There's a spike in blood glucose concentration, but then that spike reduces and then the glucose basically returns to normal. Those are the 4 different parts of that graph. If you look at a diabetic person, there's a spike, so they take the drink over here as well. There's a spike in glucose, but then that spike decreases really slowly over time compared to a non-diabetic person. And I believe that is just how you would explain that's how you would answer question 1 in this situation, because you would simply just say blood glucose concentration would rise in both of them, and then you sort of talk about the peaks as well. And you can see here that with a non-diabetic person, the peak is lower than the peak for a diabetic person. So that's something to do to just have a look at. But try to use the method that I've just used, where you split the graph into different sections so that you can explain what is happening at each section, and that should give you more marks. And then you might get, if you just say, well, a diabetic person just, has higher glucose concentration. That's already a known fact. It's telling you to describe the graph, so make sure you describe what is happening in the graph. All right, so that would give you 3 marks. Make sure to write 3 different points. Let's look at the second one. It says, name the location of the receptors in a non-diabetic person that detect a change in blood glucose concentration. So the cells that detect a change in blood glucose concentration are the beta cells. That's the islets of Langerhans, basically, or you can say in the pancreas. That would also be fine. Name the homeostatic mechanism by which blood glucose concentration is maintained. This is called the negative feedback. So always remember, negative feedback is the kind of feedback loop whereby there's a change in the physiological factor and the body, through a series of receptors and effectors, works to return that change back to its set point. So that is negative feedback, and that would be your answer there. Now, B says the urine of a non-diabetic person does not contain glucose. A person with type 1 diabetes will excrete glucose in urine. A reading of the concentration of glucose in the urine can be estimated using a dipstick. And then it tells us how a dipstick works. And I think I outlined this in my video on blood glucose concentration under homeostasis. So you can just go to playlist chapter 14 and look for the video on blood glucose concentration if you feel like you're a little bit wonky in this area. So here there's glucose in urine, enzyme A, gluconic acid plus hydrogen peroxide, and then hydrogen peroxide plus a chromogen enzyme B, and that gives us this. And then it says name enzymes A and B. Enzyme A is glucose oxidase, so that is always the first reaction in the dipstick. Glucose oxidase will react with glucose in order to form gluconic acid, and enzyme B is hydrogen peroxidase. Hydrogen peroxidase basically breaks down hydrogen peroxide in order to give us a reaction. So if you want to zoom in the details of that, because I see this video is going close to an hour now, and I'm sure some of you are just really tired. But if you want to zoom in on that, please watch the last video on blood glucose concentration, because that would be very helpful for you. Let's look at the next question. Oh yes, it says here an electronic biosensor can be used to measure the glucose concentration in a drop of blood. Suggest one advantage of using a biosensor and one advantage of using a dipstick to measure glucose concentration. So a biosensor is very quick and it's more accurate, and that is why it was developed. A dipstick, on the other hand, is non-invasive. So when you use a biosensor, you have to prick the person with a tiny needle to get a drop of blood from them, and then that drop of blood is it reacts with the enzymes on the biosensor pad, and those enzymes will then give a reading through an electric current. A dipstick, on the other hand, just requires you to dip it in a person's pee. So a dipstick is not an invasive one. And I know students usually are used to questions that ask them for disadvantages of the dipstick, but an advantage that you should bear in mind is that the dipstick does not require any blood removal, so it's non-invasive basically, and it sort of reduces the risks of you even getting a blood infection if someone had maybe used the dipstick and had not changed the needle or cleaned it or anything like that. All right, I think this is the last slide, so you can breathe a sigh of relief over here, and this is question 8, and here it says Table 8.1 shows the total number of plant species, total number of insect species, and the number of habitats in 3 areas A, B, and C. And you can see in the table there we have A, B, C. Identify the area with the highest biodiversity. So over here, let's have a look. We have one habitat. Again, I think here, the big thing here for me, as a person who's trying to answer this question, would be to first of all define biodiversity. What is biodiversity? Biodiversity speaks to the species, the number of species within a particular region, the number of habitats, the gene diversity within those species. So it's not just a matter of the number of species only, because students tend to make that mistake. They can say, okay, well, if I just look at number of species, then I should be fine. But you're looking at the region as a whole. Biodiversity also involves the number of habitats. And clearly, I mean, it's very evident here that you can see from C that you have the highest biodiversity, because C has the highest number of plant species, has the highest number of habitat species, and has the highest number of insect species. So just that would be C. And over here they say give reasons, and obviously it means you get 1 mark for area and you're going to get 2 marks for reasons over here. So a reason would be the highest number of plant species and insect species, and you also need to mention the highest number of habitats. Please don't exclude habitats when you're discussing biodiversity. Then here it says identify the area that is likely to be affected the most if the environment changes. So if the environment changes, you're going to have a big issue with the region that has the smallest number of species. This is because, again, when the species or when the population is small, you have less gene flow, so you have less variation. And that means that most of the species are probably very alike genetically. And so if there is an environmental selection pressure, they're less likely to survive because they're all the same, if you think about it that way. So diversity is good for the world. I guess that's the message I'm trying to pass between the lines. Like, diversity is an amazing thing. So over here, the area that would be the most affected would be A, because it has the least number of plant species, least number of insect species, and it only has 1 habitat. So 1 habitat, if there's a flood, then everything gets affected. State 1 reason why it is important to conserve biodiversity in all 3 areas. In this case, you can sort of talk about what is it, why do we conserve biodiversity? So we can talk about just the moral stewardship that we have. We can talk about the fact that when we conserve biodiversity, we can use them to study more and learn more about other species. We can also talk about the aesthetic reasons, so things like botanical gardens are very aesthetic, national parks and all of those things are the ways that we conserve, and so conserving them would be for aesthetic reasons. You can also talk about the fact that we maintain the genetic diversity in all 3 areas if we conserve the biodiversity. So I think I'm going to stop here. This is not the last question. I've decided to split the paper 4 video into 2 because paper 4 has another question that many students who are not familiar with past papers are usually not aware of. So I am going to put that in a separate video. It's a 15-mark question that you have to do at the end of paper 4. But until the next video, I hope you've enjoyed this one. And I hope it has sort of helped you see the areas in your in your study where you're lacking, and so that you can go back and reinforce your study methods. And it can also help you see the areas where you're really confident, and so you know where to sort of just refresh your memory quickly in preparation for the exam, and the areas where you need to concentrate. Thank you so much for watching, and I hope that you are finding these videos helpful and your friends that you invite are also enjoying them. I wish you good luck in the upcoming exams. I will try to do another video from October, November, but for now, this is what we've got. Have a good time. Goodbye.